Response is the body's ability to stay safe by affording protection against harmful agents and involves lines of defense against most microbes as well as specialized and highly specific response to a particular offender. This immune response classifies as either innate, which is nonspecific, and adaptive acquired, which is highly specific. The innate response, often our first line of defense against anything foreign, defends the body against a pathogen in a similar fashion at all times. These natural mechanisms include the skin barrier, saliva, tears, various cytokines, complement proteins, lysozyme, bacterial flora, and numerous cells including neutrophils, basophils, eosinophils, monocytes, macrophages, reticuloendothelial system, natural killer cells, NK cells, epithelial cells, endothelial cells, red blood cells, and platelets. The adaptive acquired immune response will utilize the ability of specific lymphocytes and their products, immunoglobulins, and cytokines, to generate a response against the invading microbes, and its typical features are specificity, as the triggering mechanism is a particular pathogen, immunogen, or antigen. Heterogeneity signifies the production of millions of different effectors of the immune response, antibodies, against millions of intruders. Memory. The immune system has the ability not only to recognize the pathogen on its second contact, but to generate a faster and stronger response. The inflammatory immune response is an example of innate immunity, as it blocks the entry of invading pathogens through the skin, respiratory, or gastrointestinal tract. If pathogens can breach the epithelial surfaces, they encounter macrophages in the subepithelial tissues that will not only attempt to engulf them, but also produce cytokines to amplify the inflammatory response. Active immunity results from the immune system's response to an antigen and therefore is acquired. Immunity resulting from the transfer of immune cells or antibodies from an immunized individual is passive immunity. The immune system has evolved for the maintenance of homeostasis, as it can discriminate between foreign antigens and self. However, when this specificity is affected, an autoimmune reaction or disease develops. While the immune system is meant to protect the individual against threats, at times an exaggerated immune response generates a reaction against self-antigens, leading to autoimmunity. Also, the immune system is not able to defend against all threats at all times. Transplantation rejections are immune-mediated responses, represent a hindrance to transplantation. The etiology of many autoimmune disorders is obscure. The reality is that the prevalence of these disorders increases and manifests more aggressively. Type 1 hypersensitivity disorders are immune-mediated and include allergic bronchial asthma, food allergy, and anaphylactic shock. Immunodeficiency disorders are rare, but they affect some children. Vaccination is required to induce an adequate active immune response to specific pathogens. Lev-attenuated vaccines induce both humoral and cellular response, contraindicated in pregnancy and immunocompromised states. Examples include adenovirus, polio, varicella, smallpox, BCG, yellow fever, influenza, intranasal, MMR, rotavirus, etc. Killed or inactivated vaccines induce only humoral response. Examples include rabies, influenza, injection, polio, Salk, hepatitis A, etc. Subunit vaccines. Examples include HPV types 6, 11, 16, and 18, acellular pertussis, Neisseria meningitidis, streptococcus pneumonia, Haemophilus influenza type B, etc. Toxoid vaccine. Examples include Clostridium tetani, Corynebacterium diphtheria, etc. Application concepts, cellular level, cells of the innate immunity, immune response to phagocytes, monocytes, macrophages, neutrophils, and dendritic cells, natural killer NK cells. Cells of the adaptive response are T lymphocytes, classified as CD4 T cells and CD8 plus T cells. B lymphocytes differentiate into plasma cells, which produce specific antibodies. Development. Mesoderm cells are induced to form hemangioblasts, a common precursor for vessels and blood cell formation. The definitive hematopoietic stem cells derive from mesoderm surrounding the aorta, called aorta gonad mesonephros region. These stem cells colonize the liver and after that are actively produced by the bone marrow by the 7th month of gestation. Organ systems involved. The organ systems involved in the immune response are primarily lymphoid organs, which include spleen, thymus, bone marrow, lymph nodes, tonsils, and liver. The lymphoid organ system classifies according to the following: primary lymphoid organs, thymus and bone marrow, where T and B cells first express antigen receptors and become mature functionally. Secondary lymphoid organs like the spleen, tonsils, lymph nodes, the cutaneous and mucosal immune system. This is where B and T lymphocytes recognize foreign antigens and develop appropriate immune responses. T lymphocytes mature in the thymus, where these cells reach a stage of functional competence, while B lymphocytes mature in the bone marrow, the site of generation of all circulating blood cells. Excessive release of cytokines stimulated by these organisms can cause tissue damage, such as endotoxin shock syndrome. The immune system responds variedly to different microorganisms, often determined by the features of the microorganism. These are some different ways in which the immune system acts. Bacteria depends on bacteria. Neutralizing antibodies are synthesized if the bacterial pathogenicity is due to a toxin. Opsonizing antibodies, produced as they are essential in destroying extracellular bacteria. The complement system is activated especially by gram-negative bacterial lipid layers. Phagocytes kill most bacteria utilizing positive chemotaxis, attachment, uptake, and finally engulfing the bacteria. CD8 plus T cells can kill cells infected by bacteria. Fungi. The innate immunity to fungi includes defensins and phagocytes. CD4 plus T helper cells are responsible for the adaptive immune response against fungi. Dendritic cells secrete IL-12 after ingesting fungi, and IL-12 activates the synthesis of gamma interferon, which activates the cell-mediated immunity. Viruses, interferon, NK cells, and phagocytes prevent the spread of viruses in the early stage. Specific antibodies and complement proteins participate in viral neutralization and can limit spread and reinfection. The adaptive immunity is of foremost importance in the protection against viruses. These include CD8 plus T cells that kill the man CD4 T cells as the dominant effector cell population in response to many virus infections. Parasites. Parasitic infection stimulates various mechanisms of immunity due to their complex life cycle. Both CD4 plus and CD8 plus cells protect against parasites. Macrophages, eosinophils, neutrophils, and platelets can kill protozoa and worms by releasing reactive oxygen radicals and nitric oxide. Increased eosinophil number and the stimulation of IgE by 2 CD4 plus T cells are necessary for the killing of intestinal worms. Inflammatory responses also combat parasitic infections. Despite immune response generated by intact and functional immune system, we still fall sick. And this is often due to evasive mechanisms employed by these microbes. Here are some of those. Strategies of viruses to evade the immune system. Antigenic variation. It is a mutation in proteins that are typically recognized by antibodies and lymphocytes. HIV continually mutates, thus making it difficult for either the immune system to protect against it and also hinders the development of a vaccine. By disrupting 2.5 oligoadenylate synthetase activity or by the production of soluble interferon receptors, viruses disrupt the interferon response. By several mechanisms, viruses affects the expression of MHC molecules. A virus can infect immune cells. Normal T and B cells are also sites of virus persistence. HIV hides in CD4 plus T cells and EBV in B cells. Strategies of bacteria to evade the immune system. Intracellular pathogens may hide in cells. Bacteria can live inside metabolically damaged host leukocytes and escaping from phagolysosomes, Shigella spp. Other mechanisms. Production of toxins that inhibit the phagocytosis. They are preventing killing by encapsulation. The release of catalase inactivates hydrogen peroxide. They infect cells and then cause impaired antigenic presentation. The organism may kill the phagocyte by apoptosis or necrosis. Strategies of fungi to evade the immune system. Fungi produce a polysaccharide capsule, which inhibits the process of phagocytosis and overcoming opsonization, complement, and antibodies. Some fungi inhibit the activities of host T cells from delaying cell-mediated killing. Other organisms, e.g. Histoplasma capsulatum, evade macrophage killing by entering the cells via CR3 and then escape from phagosome formation. Strategies of parasites to evade the immune system. Parasites can resist destruction by complement. Intracellular parasites can avoid being killed by lysosomal enzymes and oxygen metabolites. Parasites disguise themselves as a protection mechanism. Antigenic variation, e.g., African trypanosome, is an essential mechanism to evade the immune system. Parasites release molecules that interfere with immune system normal function. Responses, rejections, reactions, and diagnostics. Immune response. Macrophages produce lysosomal enzymes and reactive oxygen species to eliminate the ingested pathogens. These cells produce cytokines that attract other leukocytes to the site of infection to protect the body. T. The innate response to viruses includes the synthesis and release of interferons and activation of natural killer cells that recognize and destroys the virus-infected cells. The innate immunity against bacterial consists of the activation of neutrophils that ingest pathogens and the movement of monocytes to the inflamed tissue, where it becomes in macrophages. They can engulf and process the antigen and then present it to a group of specialized cells of the acquired immune response. Eosinophils protect against parasitic infections by releasing the content of their granules. Antibody-dependent cell-mediated cytotoxicity, ADCC. A cytotoxic reaction in which Fc receptor-expressing killer cells recognize target cells via specific antibodies. Affinity maturation, the increase in average antibody affinity mostly seen during a secondary immune response. Complement system. It is a molecular cascade of serum proteins involved in the control of inflammation, lytic attack on cell membranes, and activation of phagocytes. The system can undergo activation by interaction with IgG or IgM, classical pathway, or by involving factors B, D, H, P, I, and C3, which interact closely with an activator surface to generate an alternative pathway, C3 convertase. Anergy. It is the failure to induce an immune response following stimulation with a potential immunogen. Antigen processing. Conversion of an antigen into a form that can be recognized by lymphocytes. It is the initial stimulus for the generation of an immune response. Antigen presentation. It is a process in which specific cells of the immune system express antigenic peptides in their cell membrane along with alleles of the major histocompatibility complex, MHC, which is recognizable by lymphocytes. Apoptosis, programmed cell death involving nuclear fragmentation and the formation of apoptotic bodies. Chemotaxis, migration of cells in response to concentration gradients of chemotactic factors. Transplant rejection xenografts are grafts between members of different species, triggers the maximal immune response. Rapid rejection. Allografts are grafts between members of the same species. Autografts are grafts from one part of the body to another. No rejection. Isografts are grafts between genetically identical individuals. No rejection. Hyperacute rejection. In hyperacute rejection, the transplanted tissue is rejected within minutes to hours because vascularization is rapidly destroyed. Hyperacute rejection is antibody-mediated and occurs because the recipient has pre-existing antibodies against the graft, which can be due to prior blood transfusions, multiple pregnancies, prior transplantation, or xenografts. Activation of the complement system leads to thrombosis in the vessels, preventing the vascularization of the graft. Acute rejection develops within weeks to months, involves the activation of T lymphocytes against donor MHCs, may also involve humoral immune response, which antibodies developing after transplant. It manifests as vasculitis of graft vessels with dense interstitial lymphocytic infiltrate. Chronic rejection. Chronic rejection develops months to years after acute rejection episodes have subsided. Chronic rejections are both antibody and cell-mediated. The use of immunosuppressive drugs and tissue typing methods has increased the survival of allografts in the first year, but chronic rejection is not prevented in most cases. It generally presents as fibrosis and scarring. In heart transplants, chronic rejection manifests as accelerated atherosclerosis. In transplanted lungs, it manifests as bronchiolitis obliterans. In liver transplants, it manifests as vanishing bile duct syndrome. In kidney recipients, it manifests as fibrosis and glomerulopathy. Graft versus host disease. The onset of the disorder varies. Grafted immunocompetent T cells proliferate in the immunocompromised host and reject host cells which they consider non-self, leading to severe organ dysfunction. It is a type 4 hypersensitivity reaction and manifests as maculopapular rash, jaundice, diarrhea, hepatosplenomegaly, usually occurs in the bone marrow and liver transplants, which are rich in lymphocytes. Hypersensitivity reactions. Hypersensitivity reaction, a robust immune response that causes tissue damage more considerable than the one caused by an antigen or pathogen that induced the response. For instance, allergic bronchial asthma and systemic lupus erythematosus are an example of type 1 and type 3 hypersensitivity reactions, respectively. Inflammation, certain reactions that attract cells and molecules of the immune system to the site of infection or damage. It featured increased blood supply, vascular permeability, and increased transendothelial migration of blood cells, leukocytes. Opsonization, a process of facilitated phagocytosis by deposition of opsonins, IgG and C3b, on the antigen. Phagocytosis, the process by which cells, e.g. macrophages and dendritic cells, take up or engulf an antigenic material or microbe and enclose it within a phagosome in the cytoplasm. Immunological tolerance, a state of specific immunological unresponsiveness. Hypersensitivity reactions. They are overreactive immune responses to antigens that would not normally cause an immune reaction. Type 1 hypersensitivity reactions. Initial exposure to the antigen stimulates Th2 cells. They release IL-4, leading the B cells to switch their production of IgM to IgE antibodies, which are antigen-specific. The IgE antibodies bind to mast cells and basophils, sensitizing them to the antigen. When the body is exposed to the allergen again, it cross-links the IgE bound to the sensitized mast cells and basophils, causing the degranulation and release of preformed mediators including histamine, leukotrienes, and prostaglandins. This causes systemic vasodilation, bronchoconstriction, and increased permeability of vascular endothelium. The reaction can be divided into two stages: immediate, in which release of preformed mediators cause the immune response, and 2, late-phase response 8 to 12 hours later, in which the cytokines released in the immediate stage stimulate basophils, eosinophils, and neutrophils even though the allergen is removed. Type 2 hypersensitivity reactions. Antibody-dependent cytotoxic hypersensitivity, immune response against the antigens present on the cell surface. Antibodies binding to the cell surface activate the complement system and cause the degranulation of neutrophils and destruction of the cell. Such reactions can be targeted at self or non-self antigens. ABO blood group incompatibility leading to acute hemolytic transfusion reactions is an example of type 2 hypersensitivity. Type 3 hypersensitivity reactions are also mediated by circulating antigen-antibody complex that may be deposited in and damage tissues. Antigens in type 3 relations are soluble as opposed to cell-bound antigens in type 2. Type 4 hypersensitivity reactions. Delayed type hypersensitivity reactions. They are mediated by antigen-specific activated T cells. When the antigen enters the body, it is processed by antigen-presenting cells and presented together with the MHC2 to a Th1 cell. If the T helper cell has already been sensitized to that particular antigen, it will be stimulated to release chemokines to recruit macrophages and cytokines, such as interferon gamma, to activate them. This causes local tissue damage. The reaction takes longer than all other types, around 24 to 72 hours. Quantitative serum immunoglobulin. This test evaluates the levels of antibodies known as immunoglobulins present in your bloodstream. The immune system produces antibodies to combat potentially harmful bacteria, viruses, and other pathogens. These antibodies come in various types, including IgM, IgG, IgA, and IgE. IgG is distributed in both the blood and tissues. IgM predominantly circulates in the bloodstream. IgA is notably abundant in the fluids produced by mucous membranes such as saliva, tears, and nasal secretions. IgE primarily associates with immune system cells in the blood. Anti-nuclear antibodies, detection of specific autoimmune antibodies for systemic disorders, anti-dsDNA, rheumatoid factor, antihistones, anti-Smith, anti-SSA, and anti-SSB, detection of anti-RBC, anti-platelet, and anti-neutrophil, testing for organ-specific autoimmune antibodies. Microbiological studies, CRP, a marker of acute phase reactions and a sensitive indicator of tissue damage from a variety of causes. CD4 and CD8. CD4 and CD8 are two types of leukocytes found in the bloodstream. CD4 cells, also known as T helper cells, T suppressor cells, and cytotoxic T cells, aid in the body's defense against infections. CD8 cells, alternatively called cytotoxic T lymphocytes, play a role in combating cancer and intracellular pathogens. Human immunodeficiency virus, HIV, primarily targets CD4 cells. A decrease in their count and alterations in the CD4-CD8 ratio serve as indicators of HIV infection (NG) and persistent inflammation in individuals, regardless of HIV infection status. Complete blood count (CBC). White blood cells (WBCs) are a component of the CBC measured to assess immune function. Neutrophils. Neutrophils are the most abundant type of white blood cell and are often the first responders to infections. They engulf and destroy bacteria and other foreign invaders. An increase in neutrophil count, known as neutrophilia, can indicate bacterial infections or inflammation. Left shift. A left shift occurs when there is an increased presence of immature forms of neutrophils, such as band cells and metamyelocytes, in the bloodstream. Normally, the majority of circulating neutrophils are mature segmented cells. However, in response to a significant infection or inflammation, the bone marrow may release immature neutrophils into the bloodstream to help combat the infection. This shift to the left in the distribution of neutrophils is often seen in acute bacterial infections or other severe inflammatory conditions. It suggests an increased demand for neutrophils to respond to the infection. Right shift. Conversely, a right shift refers to a decreased presence of immature neutrophils and an increased proportion of mature segmented neutrophils in the bloodstream. This can occur when the bone marrow is unable to produce an adequate number of neutrophils in response to an infection or other stimulus. It may indicate bone marrow suppression or failure, often seen in conditions such as aplastic anemia, myelodysplastic syndromes, or chemotherapy-induced bone marrow suppression. Lymphocytes. Lymphocytes include B cells, T cells, and natural killer NK cells. They play various roles in the immune system. B cells produce antibodies that target specific pathogens. An increase in B cell count may suggest an ongoing immune response or antibody production. T cells include helper T cells, cytotoxic T cells, and regulatory T cells, each with specific functions in coordinating and executing immune responses. Changes in T cell counts can indicate viral infections, autoimmune diseases, or immune deficiencies. NK cells are a type of innate immune cell that targets infected or abnormal cells such as cancer cells. An increase in NK cell antibodies that target specific pathogens. An increase in B cell count may suggest an ongoing immune response or antibody production. T cells include helper T cells, indicate bone marrow suppression or failure, often seen in conditions such as aplastic anemia, myelodysplastic syndromes, or chemotherapy-induced bone marrow suppression. Lymphocytes. Lymphocytes include B cells, T cells, and natural killer NK cells. They play various roles in the immune system. B cells produce antibodies that target specific pathogens. An increase in B cell count may suggest an ongoing immune response or antibody production. T cells include helper T cells, cytotoxic T cells, and regulatory T cells, each with specific functions in coordinating and executing immune responses. Changes in T cell counts can indicate viral infections, autoimmune diseases, or immune deficiencies. NK cells are a type of innate immune cell that targets infected or abnormal cells such as cancer cells. An increase in NK cell activity may indicate viral infections or malignancies. Monocytes. Monocytes are another type of phagocytic cell that engulfs and digests pathogens. They can differentiate into macrophages or dendritic cells, which play roles in antigen presentation and the activation of other immune cells. Changes in monocyte count can indicate various infections or inflammatory conditions. Eosinophils. Eosinophils are involved in allergic reactions and combating parasitic infections. An increase in eosinophil count, called eosinophilia, may suggest allergies, asthma, or parasitic infections. Basophils. Basophils release histamine and other inflammatory mediators in response to allergens or parasitic infections. Changes in basophil count are less common but can occur in allergic or inflammatory conditions. Erythrocyte sedimentation rate, ESR. When inflammation occurs in the body, certain proteins called acute phase reactants are released into the bloodstream. These proteins include fibrinogen C-reactive protein, CRP, and immunoglobulins. In the presence of inflammation, the levels of these proteins increase, leading to changes in the properties of blood, including its viscosity. One of the consequences of inflammation is the alteration of the surface properties of red blood cells, RBCs. Normally, RBCs have a negative charge on their surface, causing them to repel each other and remain suspended in the plasma. However, during inflammation, acute phase reactants cause the RBCs to aggregate or clump together, reducing their surface charge and causing them to settle more rapidly when placed in a vertical tube, as in the ESR test. While an elevated ESR is a nonspecific marker of inflammation, it can be useful in diagnosing and monitoring various conditions, including infections, autoimmune diseases such as rheumatoid arthritis and systemic lupus erythematosus, inflammatory bowel disease, certain cancers such as multiple myeloma and lymphoma, and other inflammatory conditions. The ESR is not a specific test for any one disease, but rather an indicator of the presence of inflammation in the body. Higher than normal ESR values may suggest the presence of an inflammatory condition, infection, autoimmune disease, or certain types of cancer. However, it's important to note that an elevated ESR does not indicate the specific cause of inflammation, but rather alerts healthcare providers to the need for further investigation into the underlying cause.